The diagnostic odyssey is one of the most frustrating struggles patients and families face when addressing health issues. It begins when symptoms first occur and ends when a diagnosis is reached. For patients with rare genetic disorders, this process takes an average of 5 years. Many of these patients are children, and that time takes a toll on their developmental years. It can also be an expensive process involving numerous medical specialists and exploratory diagnostic tests, with average costs reaching over $10,000 before a final diagnosis is reached. Advancements in molecular testing are helping address these challenges. In recent years, data has shown that molecular test results have significantly improved medical management, resulting in faster access to treatment options, reduced instances of unnecessary services, and more information regarding health risks for other family members. Whole exome sequencing, or WES, is a patient-centric, phenotype-driven analysis designed to examine coding regions and splice junctions for over 20,000 genes. The results are then filtered so that only genes of plausible clinical relevance to the patient are included in the report. WES is ideal for patients with complex or rare phenotypes that have no immediate link to recognizable conditions. It is typically recommended in cases that involve intellectual disability, congenital anomalies, broad differential diagnoses, or genetically heterogeneous diseases. Compared to standard genetic testing by single genes or panels, WES has a higher diagnostic yield, is more cost-effective, and has clinical utility for active and long-term clinical management, especially for patients with nonspecific phenotypic features and critically ill neonates. The current diagnostic rate for WES is approximately 25%. While there is room for advancement, this is a significant improvement from traditional diagnostic testing. To further improve diagnostic yield, we recommend trio exome analysis to identify de novo variants, which are often causative for AD and X-linked conditions. Trio testing includes samples from the patient's biological family members to help classify variants and improve medical management. Whole genome sequencing, or WGS, is another analysis that can increase diagnostic yield. WGS helps identify repeat expansions, structural variants, regions with absence of heterozygosity, and intronic variants that cannot be detected by WES. Current research shows that WGS can improve diagnostic yield to approximately 40% and resolve approximately 5% of undiagnosed cases from WES through improved coverage, copy number variants, non-coding variants, and other factors. Full Genome is Fulgent's comprehensive whole genome sequencing test, offering analysis of over 20,000 nuclear genes plus the mitochondrial genome. It detects CNVs with greater than 2 exon resolution, genome-wide deletions or duplications over 200 kb, at least 20 repeat expansions, and clinically relevant regions of absence of heterozygosity, ensuring exceptional genetic coverage and diagnostic precision. Variant reporting is phenotype-driven to minimize reporting of variants of uncertain significance. One of the unique and powerful components of Full Genome is the ability to integrate with RNA analysis for functional insights into genetic variants, enabling deeper characterization of pathogenicity. Based on the statistics we've discussed so far, professional institutions like the American College of Medical Genetics and Genomics, as well as the American Academy of Pediatrics, recommend WES or WGS as a first-tier test for developmental delay, intellectual disability, and congenital anomalies. Additionally, the National Society of Genetic Counselors recommends WES or WGS for cases of unexplained epilepsy, a recommendation endorsed by the American Epilepsy Society. While the benefits of WES and WGS are many, there is always room to improve, and that is why Fulgent has developed RISE. RNA-Integrated Sequence Evaluation. RISE combines WES or WGS analysis with transcriptomic analysis by RNA sequencing. This enhances the diagnostic yield of complex or undiagnosed cases by assessing the RNA-level impact of coding regions, splice site changes, and variants in non-coding regulatory regions of phenotypically relevant target genes. Variant prioritization and interpretation are among the main challenges of genomic testing. RISE addresses this by adding additional information through RNA, which provides a more dynamic perspective than DNA and can help identify abnormalities that would not be picked up by DNA alone. When we look at RNA in the lab, we perform multiple analyses in one test. As with most competitor analyses, this includes splicing events, but Fulgent's RISE also evaluates aberrant and monoallelic gene expression. Looking more closely at these analyses, aberrant gene expression is when expression significantly deviates from the normal physiological range. This is typically accompanied by low protein levels, which may be related to a genetic disorder. Transcripts with nonsense variants are frequently degraded via nonsense-mediated decay, which may be detected by aberrant underexpression of genes. Nonsense variants often result in premature termination codons. We're able to interpret them in DNA with corresponding RNA analysis for confirmation for classification purposes. However, these analyses provide information in more challenging situations, such as for variants in non-coding and regulatory regions such as promoters, enhancers or suppressors, variants in the untranslated or intronic regions, or large deletions that have the potential to cause aberrant underexpression of disease-causing genes. Monoallelic expression, or MAE, is when one of the two alleles is primarily expressed, while the second is only expressed at very low levels. MAE is a specific form of aberrant expression and an extreme form of allelic imbalance. It may escape detection by aberrant expression studies, since expression of mainly one allele does not always provide overall expression levels outside the normal physiological range. MAE can be related to epigenetic silencing, such as in the cases of X inactivation or genomic imprinting. We use RNA sequencing to count the reads aligned to each expressed allele at genomic positions of heterozygous single nucleotide variants to detect monoallelic events. The final component of our RNA analysis is aberrant splicing. At least 10% of pathogenic variants have an effect on RNA splicing. Canonical splice sites are very strong pathogenicity based on ACMG criteria, but deep intronic and exonic or synonymous variants that have an impact on splicing are harder to predict. RNA sequencing addresses this shortcoming by quantifying splicing events by detection of split reads whose ends align to distinct sequence elements. With RNA-Seq, we can detect exon creation, exon skipping, exon truncation or elongation, or intron retention. Thanks to the expansive data provided by both DNA and RNA analysis, RISE transcriptomics increased diagnostic yields by approximately 17% over traditional WES or WGS testing. When analysis is complete, we'll provide an easy-to-read report with clinically significant variants clearly labeled on the front page. In cases where we identify something in the RNA that does not have a corresponding DNA variant, we will also include information regarding monoallelic expression or similar findings in this box to ensure you receive comprehensive, clinically relevant data. RISE is validated for blood specimens only. You have the option of singleton or proband-only testing, as well as the preferred duo or trio option. Please note that RNA sequencing is only performed on the proband when we receive duos or trios. Turnaround time for this test is between 6 to 8 weeks for WES and 3 to 5 weeks for full genome. To sum up, RISE offers a richer analysis than WES or WGS alone, thanks to the addition of RNA sequencing. While many labs only use this data for the reclassification of qualified variants related to splicing, at Fulgent, we provide additional information on allelic-specific expression or imbalance and differential gene expression. With this data, we can identify the impact on gene expression levels from regulatory regions, the functional consequences of splice site variants, and large insertions or deletions and complex structural changes. We can also confirm structural variants, including tandem duplications, gain insight into imprinting disorders like UPD, determine the status of skewed X inactivation, and verify and distinguish variants from sequencing artifacts and pseudogenes. All of these insights would likely be missed when analyzing DNA alone. For more information about RISE and full genome, visit fulgentgenetics.com. If you have any questions, feel free to reach out through info@fulgentgenetics.com. Thank you.